Hello, my dear students. Welcome to my today's online video lecture on PCR technique, that is polymerase chain reaction. It's a technique which has a wide applicative value. Today we are going to learn about this technique, how exactly it is carried on. So, I am Dr. Meera Beke, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. There is my cell phone number and mail ID on this slide, and by now you must be knowing why they are there. After the class, any queries, do contact me. Now, you can see an automated machine picture in the slide. That is actually the PCR machine. This PCR is carried out in an automated machine, and different models are available, manufactured in different companies, and this is one such typical PCR machine. Now let us see what exactly is done in PCR. PCR is an in vitro technique for generating large quantities of a specified DNA. Please note, it is in vitro, means this is done in laboratory, not inside the cell systems. And the main purpose is to generate large quantities of a specified target DNA of your choice. It is just to have multiple copies of a specified piece of DNA. Something like xeroxing the material what you want multiple times, 100 copies, 1000 copies, like that. Here you will generate large quantities of specified DNA. So the moment I tell it is in vitro technique, I mean to say it is a cell-free amplification technique for synthesizing multiple identical copies of any DNA of interest. So billions of copies are generated within a short time. That is the significance of PCR. Now let us see when it was discovered and by whom. The technique was developed by Carey Mullis in 1984, and he was awarded Nobel Prize for this in the year 1993. Now, PCR is considered as a basic tool for a molecular biologist. The way you use your laptops, the way you use your Xerox machines in the offices, in the same way, for a molecular biologist, PCR is something like that. Okay? Now, what is the principle? The double-stranded DNA of interest is denatured to separate into two individual strands. Okay? The double-stranded DNA of interest is denatured. The first step in PCR reaction is denaturation. Okay? Denaturation. Then, each strand, that is single-stranded, is then allowed to hybridize with a primer. This is called renaturation. Then, the primer template duplex is used for DNA synthesis using the enzyme DNA polymerase, and this step is known as synthesis. So basically, in any PCR reaction, three steps are involved: denaturation, followed by renaturation, followed by synthesis. Okay? So, denaturation is separating the two strands of double-stranded DNA of interest. Then, hybridization of each strand with the primer is called renaturation. And once you get a primer template duplex, if it is used for DNA synthesis, In presence of the enzyme DNA polymerase, it is called synthesis. So these three steps, denaturation, renaturation and synthesis, are repeated again and again and again to generate multiple copies of target DNA. This is exactly the principle of PCR. Then what all you need to conduct a PCR? First and foremost thing is you need a target DNA whose multiple copies need to be generated. This target DNA is normally 100 to 35,000 base pairs in length. However, shorter the target DNA, efficiency of PCR is more. Okay. Then, the second essential requirement happens to be the primers. You need 2 primers. What do you mean by primers? See, these are synthetic oligonucleotides. Automated machines are available to synthesize these oligonucleotides of known base sequence. Okay, you can synthesize them. Normally, they are of 17 to 30 nucleotide length. 17 to 30 nucleotide length, and they are complementary to the regions flanking the target DNA. So they are complementary to the regions flanking the target DNA. Since you know the target DNA of your choice, and on either side you know what bases are present, and accordingly you can always synthesize the oligonucleotides that need to be used as primers. Okay, and please note, they should be present in excess always in the reaction mixture for efficient PCR to take place. Okay. Then you need, of course, deoxyribonucleotides, which are the building blocks of DNA, that is DATP, CTP, GTP and TTP, right? These are the 4 building blocks or monomeric units which go into the formation of DNA molecule, right? Then most important thing is you need thermostable DNA polymerase. Our regular DNA polymerase cannot be used here. What we need is thermostable DNA polymerase that can withstand a temperature up to 95 degrees Celsius. The enzyme should be able to withstand a temperature of 95 degrees Celsius. Okay. Basically, it's a protein, right? So, normally at that temperature, proteins undergo denaturation. It should not happen, right? And normally, we use Taq polymerase. Taq polymerase is a thermostable DNA polymerase which is isolated from the bacterium Thermus aquaticus. Thermus aquaticus. Okay. There are other thermostable DNA polymerases. I will let you know little later. Okay. And the drawback of this particular Taq polymerase. This technique involves repeated cycles for amplification of the target DNA. Each cycle has 3 stages, right? Each cycle has 3 stages. Yeah. This is the schematic representation, right? So, the green colored ones are the original DNA to be replicated. We need multiple copies of this particular DNA. And the red ones represent the DNA primer. And the blue ones are the nucleotides. Okay? ATP, GTP, CTP and TTP. Right? So, in the first step, what is done, as I have already told you in the principle, denaturation should take place. When you raise the temperature, say to around 95 degrees Celsius, okay, for about 1 minute, just 1 minute, the 2 DNA strands get denatured and the strands will separate out. You can see in the diagram, the 2 strands have separated out. This is the first step, that is denaturation. This is followed by the second step, that is renaturation, or it is also called annealing. And you should cool the mixture now. Okay, as the temperature of the mixture is slowly cooled to about 55 degrees Celsius. From 95 to, you should cool it to 55 degrees Celsius. The primers, the red colored ones, base pair with the complementary regions flanking the DNA strands, target DNA strands, and that is what you can see in the step 2. And this process is called renaturation or annealing. And in the reaction mixture, you should see that there is a higher concentration of primers. Okay, this ensures annealing between DNA strand and the primer rather than the two DNA strands. So when you maintain higher concentrations of primers, the two separated DNA coming together is avoided. That's the purpose why you should have higher concentration of primer strands. This is the second step in PCR, that is renaturation. This is followed by synthesis. You can see the third step here. The initiation of DNA synthesis occurs at 3' hydroxyl end of each primer. You can make out here. So the original DNA strand 5' and 3' and opposite 5' and 3', but the synthesis begins at the 3' hydroxyl end of each primer, both the sides. You can see how exactly the reaction takes place. The primers are extended by joining the bases complementary to DNA strands. Already you have given all the 4 bases, DATP, DGTP, GTTP, right, and DCTP, right. All the 4 are available. Based on that base complementarity rule, the DNA strands are extended. This synthetic process is quite comparable to the DNA replication of the leading strand. Now, I want you to go back to your previous semesters and remember about DNA replication. There you must have studied, right? Leading strand and lagging strand, Okazaki fragments, all that, right? So here, the synthesis is almost comparable to the DNA replication of the leading strand. So as a homework, you can just go back to your molecular biology notes and learn more about DNA replication of the leading strand, not the lagging strand. Okay? Now, for the reaction to take place, the temperature has to be kept optimal as required by the DNA polymerase. For example, the optimal temperature for Taq polymerase is around 75 degrees Celsius. So, the synthesis will take place, and if you want to stop, you have to just do one thing. You can stop the reaction by raising the temperature to about 95 degrees Celsius. That's all. So each cycle of PCR takes just 3 to 5 minutes. Just 3 to 5 minutes. So in normal practice, the PCR is carried out in an automated machine that I have shown you. Denaturation 1 minute, temperature approximately 95 degrees Celsius. Annealing or renaturation about 1 minute, temperature approximately 55 to 68, and synthesis approx 2 minutes, temperature approximately 75, right? So this is what exactly happens during the PCR. Now, the new DNA strands are formed, right? So the new DNA strand joined to each primer, what has been shown in blue color in this diagram, is beyond the sequence. Please note, is beyond the sequence that is complementary to the second primer. These new strands, the blue ones, are called long templates and they will be used in the second cycle. Blue ones are called long templates and they will be used in the second cycle. Okay. So this is what exactly happens in PCR. Let us see some more details in next slides. So same thing, whatever I have explained, I have given here. So denaturation 95 degrees for about 1 minute. Then renaturation 55 degrees. So all this pairing and higher concentration I have told you, right? Then here you can see another PCR schematic representation wherein I have explained the synthesis, right? Then, so this is the first step. Then DNA synthesis by Taq polymerase. Then you will get duplicated DNA fragments. So at the end of cycle 1, PCR cycle 1, you get 2 copies. At the end of cycle 2, you get 4 copies. At the end of 3, you get 8 copies. That is a matter of 15 minutes. So like this, you keep on getting, finally you get billions and billions of copies, right? So same thing I have repeated here. Now, so I have given the timings. Yeah, this is the graph showing you the time taken for each step and the temperature required, right? So the new DNA strand joined to each primer is beyond the sequence that is complementary to the second primer. So these new ones are called the long templates which are used in the second cycle. So for the second cycle of PCR, the DNA strands original 2 and newly synthesized long templates 2 plus 2, 4 are subjected to denaturation, annealing and synthesis, right? So at the end of second round, you will get long templates and the DNA strands which you get in the second cycle on long templates which are coded along the long templates are called short templates, right? That is DNA strands with primer sequence at the other end and sequence complementary to end primer. These are formed. So at the end of second round, you will get virginal strands, long templates and short templates which are used in the third cycle of PCR. In the third cycle, the virginal DNA strands along with short and long templates are the starting materials. Denaturation, renaturation and synthesis are repeated. This procedure is repeated again and again and it is estimated that at the end of 32nd cycle of PCR, how much time? 32 into 5, okay? So 32nd cycle of PCR, about 1 million-fold target DNA is synthesized within such a short span of time. Okay, this is the beauty, significance of PCR. Now, when you are doing all this, what are the key factors that you need to remember to conduct PCR at optimal condition so that the products are ideal? Okay, now primers. See, you have to have the primers of 17 to 30 nucleotide length and it is preferable to have primers without secondary structures. They should not have any loops. That's what I meant by secondary structures. And they should be without complementarity among themselves. Otherwise, these primers only will join to each other where the whole reaction goes a big waste. So coming to primers, you should have 70 to 30 nucleotide length. They should not have any secondary structures and they should be without complementarity among themselves to prevent auto matching, right? Then DNA polymerase, I was telling you. Normally we make use of Taq polymerase isolated from Thermus aquaticus, but there is one drawback with this enzyme. It lacks proofreading exonuclease activity, 3' 5' exonuclease activity. That means normally any DNA poly will have this proofreading exonuclease activity. By chance, if a wrong nucleotide is added, it will proofread and it will remove the wrong nucleotide followed by the addition of right nucleotide. And unfortunately, this Taq polymerase do not have that capacity of proofreading and removing wrong nucleotide by chance if it is placed. So this may lead to errors in PCR products. So nowadays, people use TMA DNA polymerase, which is isolated from Thermotoga marittima bacterium again, Thermotoga marittima. So TMA DNA polymerase is used, and another DNA pol of our choice. All the three are thermostable. PFU DNA pol or PFU DNA polymerase isolated from Pyrococcus furiosus. Okay. So of the three you can use any one. Taq polymerase has this drawback of lacking proofreading exonuclease activity. Then target DNA. I told you, shorter the target DNA, better is the efficiency, right? And the sequence is also of paramount importance because if the target DNA has too many GC components, okay, GC rich DNA strand hinders PCR, right? So the length is of importance and the sequence is also equally important, okay? So this is about the target DNA. Then promoters and inhibitors of PCR. See, there is a chemical called BSA, bovine serum albumin. If you add BSA to the reaction mixture, it will enhance the PCR efficiency. On the other hand, if you add humic acids, that will inhibit the PCR. So, if you want to have PCR in optimal conditions, you have to take care of all these things, all right? So, this is all about the PCR technique. Dear students, thank you for listening, and in my next lecture class, I am going to talk about the applications of PCR. Okay, it has a wide application as on now. I am going to discuss in detail in my next class. Thank you for patient listening. Stay blessed. Any queries, do contact me.